Hey, Bio30s, and welcome to our discussion today about assistive reproductive technologies and contraception. In past videos in this unit, I've been discussing how males and females produce gametes in their respective reproductive systems, how those gametes come together to fertilize a zygote, which would then go on to become a viable embryo, fetus, and then be born into a fully fledged human. However, there are some individuals that have issues conceiving children, and they require what are called assistive reproductive technologies or ARTs to assist them. Generally speaking, these are either going to be people that are sterile, who have absolutely no way to conceive a child whatsoever, biologically speaking, that is, or those individuals that either one or two of the respective partner, or I guess male or female, have an issue with infertility. And that is more common where they've been trying for perhaps a period of a year, consistently trying to produce a child, and have had no luck in doing so. Then they might be considered to be infertile, either the male or the female, or both could be infertile at that point. What are some of the reasons why males or females can become infertile? Well, we've discussed a couple of these already, but let me quickly go through them again. If you're a male, you could have a blockage or damage to your epididymis or your vas deferens, which are going to transport your mature sperm cells toward the ejaculatory duct. That could be an issue. You can also have a low sperm count. We've talked about that already in the male reproductive system video. A count below 20 million sperm per milliliter. This can be, the risk factors for this can be smoking, excessive alcohol use, or overheating when the sperm are being produced. Something that I didn't mention in the male reproductive system video is that the male's gonads, the testes, are basically hanging or dangling below the main body cavity for the reason that sperm require a slightly cooler temperature to actually undergo spermatogenesis than body temperature. They prefer a temperature of around 35 degrees. So that's why the male's genitalia, or I guess the gonads, are dangling below the main body cavity rather than deep inside the body cavity, like in the case of females where their ovaries are. So that can be an issue. A temperature when the sperm are produced can also be an issue. You can also have abnormal sperm. You might have normal sperm counts, but if the sperm themselves are not fully functional, and I've also discussed this in the male reproductive video as well, and it's in the notes if you go back to the reproductive notes. Risk factors here could again be exposure to toxins, STIs, or overheating during spermatogenesis. I think I give you some examples of this where the tails aren't long enough, maybe the tails are bent, the heads aren't big enough, or the heads are too big, or they have multiple heads or multiple tails. There's issues with more than the average amount of sperm that are not normal, and that can be just as bad as having a low sperm count. You can have a high sperm count, but if most of your sperm are not fully functional, then that can also be an issue as well. And then finally, there can also be the issue of impotence, where males cannot become aroused during sexual intercourse. Risk factors here can be caused by a number of factors. Vascular disease can be an issue. Nervous system injury, stress, hormonal imbalances, medication, smoking, and alcohol consumption can all be factors when it comes to impotence in males during sexual intercourse. So these are some of the reasons why males can be considered to be infertile. Females, on the other hand, can have issues including they can have a blocked oviduct. If their oviducts are blocked, then the sperm can't get to the egg, or vice versa. This can be caused by this could be even caused by an STI, like we discussed in a previous video, actually the last video, or 2 videos ago when we discussed that. If women can't ovulate, that's obviously an issue as well. This can be caused by hormonal imbalances, malnourishment, or stress in their life. You can also have something called endometriosis, which is a painful condition in which the endometrium will actually grow outside of the uterus rather than on the inside of the uterus. So if there is no endometrium inside the uterus to receive a fertilized egg or a zygote, or at that point it would be an embryo, then the embryo has nowhere to basically plant itself in the uterus, and then you can't conceive a child. So that can be an issue as well. And there can also be damage to the eggs that can be a factor as well. Risk factors here, again, are going to be exposures to toxins and or radiation. So those are a few of the reasons why a female can become infertile as well. So it can happen on both sides. So what are some of the solutions to these issues of infertility or sterility? Well, there's a few types of assistive reproductive technologies, and I'll kind of go through a few of them here just to kind of let you know what they're all about. First one is called artificial insemination, AI, or intrauterine insemination. This has been around for decades, and basically they're going to collect sperm, and they're going to take the sperm and they're going to basically put them directly into the uterus, deep inside the female's uterus, to give these sperm a fighting chance to get closer to the oviduct, so that more sperm can get to the oviduct and hopefully make contact with an egg that's being released into the oviduct. This way, you're avoiding the sperm having to enter the vagina, the cervix, a majority of the uterus. They're basically placed as close as they can up toward the entrance of the oviducts, so the sperm can actually get to where they need to go. This can be also used for females that don't have a male partner. So if they want to conceive a child without a male in their life, they can basically just go to a sperm bank, get the sperm, and then undergo this procedure where they can be artificially inseminated to basically conceive a child. But this has been a process that's been around for a while. In even more, I guess, to increase the odds even more, we can use something called IVF, or in vitro fertilization. This is where we take an egg out of the female, sperm out of the male, and we basically put the two of them together, and we fertilize them in the lab, essentially in a petri dish. We basically combine them together, and in some cases, you can even inject, as you can see in the image here, you can even inject the male's genetic material from the sperm directly into the egg to basically seal the deal. These eggs, after fertilization, then several of these embryos can then be implanted into the female, and hopefully at least one or maybe even two of them, or maybe even more, might take inside the female's uterus, and then they basically will get implanted. And again, the timing is very important here. They got to make sure that they have to time it so that when the woman's endometrial lining is at the highest or thickest level. So around day 21 of the menstrual cycle would be kind of when they would need to be implanted after they've been fertilized in the lab in this IVF procedure. There is a slight variation on IVF that can be used as well. It's called GIFT. It's called gamete intrafallopian transfer. Basically, you're going to take the eggs and the sperm, and you're going to basically combine them together directly in the oviduct, and hopefully that this is actually going to occur naturally inside the female rather than in a petri dish in the lab. So that's what GIFT is when we basically inject the sperm into the oviduct directly and try to make contact with an egg in the fallopian tube rather than it happening inside of a lab environment. So those are those. You can also use surrogate mothers. If the mother, let's say the mother has an issue with her uterus, maybe she has had a hysterectomy, doesn't have a uterus, or has maybe endometriosis, it can't actually, she can conceive a child, but she can't actually bring the child to term. So a surrogate mother can be contracted by an infertile couple to basically carry the baby for them. You can use AI or you can use IVF to basically bring the one or the two gametes together, and then they can basically inject the embryo into the surrogate mother, and then that mother would then carry the baby to term, and then would then give the baby to the parents once the pregnancy is over. Some women who have issues with ovulation can undergo a procedure called superovulation, which causes them to release multiple eggs at the same time because of hormonal treatments. This technique is often used in conjunction with other assistive reproductive technologies. So this is going to allow a woman to basically produce multiple eggs, and hopefully one of the eggs will take. There is a risk here. Obviously, if you have a woman who's ovulating 4 or 5 eggs at the same time, what if all 4 or 5 of the eggs get fertilized all at once? Now the woman is carrying potentially 4 or 5 babies all at once, fraternal twins or fraternal quintuplets in this case. There is issues with that. So I mean, the doctors who use this technique need to be very familiar with the patient and understand, and the female who's undergoing the procedure should also be very familiar with the risk that if you're trying to conceive one child, what happens if you conceive 3 or 4 children? That's a huge commitment if you're just wanting one and you get 4 babies. So there are some issues there with that. And there's health issues as well and financial issues, raising the children after they're born and stuff like that. One area of reproductive technology that I want to discuss. It's not one that's used in humans, and thank goodness, but there is cloning. Cloning is another form of reproductive technology that's out there. You may have heard of, Dolly the sheep, who got cloned in 1986, if I'm not mistaken. And there's been a lot of, people who can pay to get their pets now cloned after their pets die. This is stuff that people can get done, and there's obviously, there's big money for companies to do this because people want to have their pets back or whatever. The cloning has been around for millennia. I mean, the plants have been cloned for centuries now. Grapes have been cloned, in Europe for millennia. Sorry, I said that Dolly the sheep was cloned in 1986. It was 1996. Pardon me. It was 1996. Dolly was cloned. The sheep. Interestingly enough, that clone took 433 failed attempts before they actually got a clone to actually survive. That's not something that's not normally commonly discussed in the news reports about Dolly, but there were hundreds of failed attempts before they got Dolly to actually work. There has been a lot of talk now about cloning to resurrect extinct species. Obviously, Jurassic Park, which is a pipe dream, that's never going to happen. DNA can't survive that long. Hate to tell you, we're never going to see T-Rexes again. But we could see more recently extinct species that have DNA intact. Tasmanian tigers, even a woolly mammoth, perhaps. There have been some woolly mammoths that have been basically survived in the Arctic. Like their DNA and their tissue samples are sort of intact because they were frozen in the permafrost. So there are some discussions about the use of cloning to resurrect extinct species. If you're interested in learning a little bit more about cloning, watch the video here. Some interesting stuff, and again, some ethical issues there. Should we be bringing back extinct species that had their shot, or maybe died out a few thousand years ago? Should we be bringing them back into a modern ecosystem that doesn't really know what these animals are? Would they be considered to be maybe an invasive species now that they're gone, kind of thing? So, but again, watch that video. There's some interesting points that are being brought in there. Cloning humans in the future, dangerous territory. This is sort of what we talked about with we talked about germline stem cell research and modifying and making custom-made babies. Cloning humans would fall into that category. There's a lot of controversy, and obviously there's a lot of loss right now related to germline manipulation of embryos, and cloning would fall into that category as well. All right. Well, not everyone has trouble conceiving. There's a lot of people who don't want to conceive. There are people out there that they're not ready to have a child. They want to practice they want to have sex, but they don't want to have the ramifications of perhaps having a child afterward. So for those individuals, and for a lot of people out there that are doing this, this is what we call contraception. Contraception is any technique that's going to prevent you from basically getting pregnant. But not only that, some of these contraception techniques are also going to help prevent or very much minimize the possibility of getting an STI, like we discussed earlier in the unit. So there are many ways that you can there's many methods of contraception. The one that is 100% effective all the time at both preventing pregnancy and STIs is abstinence, where you don't have sex at all. And that's basically going to work because you can't really have a child if you don't have sex, right? You can't get an STI if you're not having sexual relations. So that's one way that's 100% effective. But there are other methods out there that can be used that have high rates of success. There's really no 100% contraceptive technique, but there are some that are much more effective than others. And I will go over which ones are considered to be the most effective methods of contraception and the ones that are most effective at reducing STIs as well. So there's a couple of ways you can, so the one way you can do it is you can do surgical. There are some people who are just, okay, I'm done. I don't want to have any kids anymore. I just want to basically not have to worry about the hardware down there working anymore. So you can have surgical procedures or sterilization procedures that will work for both a male or a female. In the case of a female, we have what's called tubal ligation, where you basically either snip or tie the oviducts, and that way the eggs that are being released by the ovaries have nowhere to go. The sperm and the eggs are basically separated by a barrier that they can't actually cross over, and then if you can't get the gametes together, then basically you can't have a child. In a male, we have what's called a vasectomy. That's where you actually you snip or tie the vas deferens of the both vas deferens of the male, the vas deferentia, if you want to say the plural, or the ductus deferentia, if you want to say the other name for it. But basically, that's going to get sort of tied or cut in the male, and then they basically can't have any children. Interesting thing about a male having a vasectomy, there is about if you're ever going to have this in the future, and your doctor would probably talk to you about this, but after you've had a vasectomy, you actually have about 20 to 30 ejaculations after that to clear out any remaining sperm that are inside the viable section of the ductus deferens that's left over. So once you have the vasectomy, you're still reproductively viable for about 20 to 30 ejaculations after that. Then at that point, once all the sperm have cleared out the remainder of the vas deferens and the ejaculatory duct, then basically at that point, your gametes aren't going anywhere. All you're doing is you're producing semen, and no sperm are entering. You would be producing all the fluids from your prostate gland, your seminal vesicles, and your Cowper's gland, but there would be no sperm as part of that semen mix that is being ejaculated at that point. There are some pill methods that can be used, both in this case for women. Birth control pills in women have been around for a long time. It basically raises levels of estrogen and progesterone in the body. This exerts a negative feedback loop on the hypothalamus, which is going to stop the production of GnRH, which then basically stops the production of FSH and LH. And if you don't have FSH and LH, you can't ovulate. So basically, there's also no follicular development. So they basically artificially keep the levels of estrogen and progesterone really, really high to basically force the female not to basically release FSH, which is going to trigger the follicular phase of the ovarian cycle. The pill is taken for 21 days of the 28-day menstrual cycle. The last 7 days, they have to stop taking the pill because we obviously want the progesterone and estrogen levels to drop in order for the female to menstruate. Once menstruation has occurred, the female then goes back on the pill for the remaining 3 weeks. This is going to stop the development of the follicle during the follicular phase, days 0 to 14, and then will prevent any kind of issues up to the potential implantation of the embryo, and then at day 21, off the pill, let the menstruation occur, and then start all over again. The morning after pill is another pill that can be used after sexual intercourse. It's several pills that can be taken within 1 to 2 days of an intercourse event. High levels of estrogen and progesterone are at the key of it as well. This disrupts the ovarian cycle. This will also prevent implantation or ovulation from occurring. The effectiveness of the pill definitely drops dramatically as you go further away from the intercourse event. If it's taken within the first day, it's 95% effective. After 2 days, it's 85% effective. And on the third day, it's only 5% effective. So this is definitely a pill that, as the name implies, you take sort of the morning after a sexual encounter. The most effective method, and this was determined as the most effective form of birth control for females, birth control in general. In 2008, it was the Canadian Pediatric Society that came up with this in 2018, and they said that this method of birth control is 99% effective. And this is called an IUD. It's called an intrauterine device. So this device is inserted into a uterus for up to 12 years. This is actually placed inside of a female, and it basically releases hormones that thicken the mucus around the cervix, so there's a cervical plug that the sperm can't get in. It also stops ovulation, and they're made of copper, which raises the white blood cell count in the uterus, which is going to be more effective at destroying any gametes that get in there. So basically, this is a long-term method of contraception up to 12 years inside the female, but proven to be very, very, very effective indeed. You can also get what are called birth control shots. There's what are called the Devo Provera birth control shots. They're taken every 12 weeks, and they release a hormone similar to progesterone, which helps again stop FSH release, so there's no follicular development. You can also get what's called a progestin implant. This is the size of a matchstick, and it's placed under the skin of the upper arm, and it can last up to 4 years. And this stick basically releases the equivalent of progesterone and estrogen. Again, the same idea as a birth control pill, but it's basically releasing it on its own without you having to worry about taking the pill every month. This is basically, it just automatically releases the hormone levels that we need to release, and then it just takes care of it. And again, up to 4 years, it can be under the skin. Physical barriers are very effective as well. These are probably the most common, well-known versions of contraception in males. There's both male and female condoms, which are basically I guess penis-shaped or vagina-shaped barriers that are basically create a physical barrier between the penis and the entrance to the cervix. The gametes can't get into the female's reproductive system. These are the most effective method at preventing STIs. They may not be as effective. They're definitely effective for birth control, but not as effective as an IUD. But all of these methods up here that I've talked about, surgical sterilization, pills, IUDs, the shots, the implants, none of them will help prevent STIs. Condoms are by far the most effective birth control method for preventing STIs if you want to prevent getting them during sexual intercourse. You can also have what are called contraceptive caps. These are inserted inside of the female, basically to the back of the vagina, up against the cervix. You can have a diaphragm, or you can have what's called a cervical cap. The main difference between them is the diaphragm tends to be a bigger diameter than the cervical cap, but both of them provide a dome shape cover over the cervix, which prevents the gametes from getting into the cervix and going up toward the uterus at that point. You can also use, in combination with this, you can also use what is called spermicidal jellies, which are going to basically interact with the sperm and help to destroy them or neutralize them. These caps, just to be aware, they shouldn't be left in the vagina for more than 30 hours at a time. They should be removed, fairly soon after intercourse, but they can be kept in for as long as 30 hours. But normally, I think that they would want to be removed fairly shortly after sexual intercourse, for sure. And then the last method, which is sort of, I don't know, to me, it isn't going to help with STIs. It's not really, and again, if the female's menstrual cycle isn't exactly where it is on the 28-day cycle, and remember, women, their menstrual cycles will vary month to month depending on factors in their life, stress, diet, that kind of stuff. You can use what's called a natural family planning or a rhythm method. They have to be very aware of what's going on with the female. This method is probably not the most effective. These methods here are probably going to be a little bit more proven results if you're at least taking the medications or the pills when you're supposed to, or you've got the device, or you've got the surgery done, or you're using a condom during intercourse. Those are more kind of concrete ways to prevent these things from happening. Little biological accidents from happening. Here's a recap video. We've watched the part 1 and part 2 videos of the reproductive system, where Hank discussed the male and the female reproductive systems. This is going to be discussing the main event, the actual sex, the fertilization, which we discussed last day. He'll discuss that again, plus some methods of contraception and stuff like that. For homework, not a lot, but we do have a little bit. You've got a booklet called Arts and Contraception question booklet. That's the one you want to be going through, covering all the topics that we discussed in today's lesson. Thanks for tuning in. Next time I'll be discussing the beginning of embryonic development. Well, actually all of embryonic development. I'll be discussing weeks 1 to 8 in prenatal development, where a lot of stuff is going to be happening in a very short amount of time. Thanks for joining me, and I will see you next time.